Hey everybody, welcome to week 4 of physiological psychology here from Dr. Kirby's living room. We will be talking this week about the smallest units in the nervous system, which are neurons and their supporting cells, glia. So let's get started with that. I sort of rearranged the readings in this class. Normally I used to teach this class with this chapter first, chapter 1, but I found that students considered this pretty abstract. They didn't understand why these tiny cells are very connected to psychology, and they had a hard time remembering a lot of this dense biology information we're going to have to memorize for this chapter. And there's a little bit of chemistry involved. It can be a little tricky. So it sort of discouraged students when we put it at the very beginning. So I wanted to zoom out and start the class with a little more big picture ideas and explaining what biological psychology is and how it's connected to psychology. Remember, for this week, you need to read, instead of a bunch of broken up modules this time, all of chapter 1 in your textbook. You'll also need to complete collaborative quiz 3 and prep guide 3, for which there will be an attached article to read as per usual. We're going to talk about types of nerve cells, glia, structure and function. So that's glia is one of the types of nerve cells. Neurons are the other. We'll talk about their structure. They also have kind of a complicated function, and we're going to talk about the detailed steps of that in terms of resting potential and action potential. And then we're going to end with the blood-brain barrier. So just keep that in mind when you're doing your reading if you're confused about where to look to kind of follow along. Remember that the nervous system has many different branches. We start with the central and peripheral dichotomy. The central nervous system is split up into the brain and the spinal cord. The peripheral nervous system is split up into somatic and autonomic nervous systems. The somatic helps us more with conscious movements and sensations that we're aware of. Our autonomic nervous system is the one operating mostly outside of conscious awareness and control, and it's split up into sympathetic or fight or flight and parasympathetic or rest and digest. But instead of zooming in on any particular part of this, like we have sometimes done in previous chapters, or talking about how they develop or anything, we are going to zoom in specifically on neurons, the tiniest building block that builds up each one of these systems. We have neurons and glia. Neurons are a lot bigger than glia. They are a bit less plentiful, so there are 10 times more glial cells in your nervous system than there are neurons, and you already have around 86 billion neurons, so the number of glia is just astronomical, hard to even comprehend. Neurons have the very important function of sending and receiving messages. What do we mean by messages? I'm going to get into that much later in this lecture series, so don't worry about that just yet. Glia serve a variety of supporting functions. Early scientists called them glia, which is the Latin word for glue, because that's what they thought they did. They just were sticky stuff that helped the neurons stay in place. But actually, they serve a lot of a variety of very important functions without which the neurons could not survive and conduct their messages. Glia can have many different structures. Here we're looking at them through an electron microscope photograph of 2 types of glia. We're going to look at a few more types on diagrams. After this, we're going to use primarily diagrams, because on the actual photos it's really hard to see and understand the different structures we might need to study. This is just an example of their variety in shape. On the left we see several vaguely star-shaped astrocytes, and on the right in panel B, this cell looks more like a kind of tangle to me, like a ball with little root-like tendrils coming off from all sides in a really ragged way. This one is an oligodendrocyte. Now let's move on to diagrams of them that are easier to study and talk about what they do. Here we have a 5-panel figure with each panel displaying a different type of glia. In panel A on the top left, we see an astrocyte. It's vaguely star-shaped, like we saw on the previous electron microscope photograph. Here we can see that its little branches are attached to capillaries, which are tiny blood vessels, and also to neurons. These cells nourish neurons and carry away their waste. In panel B, we see microglia. This one is very small relative to the neurons, and it has more and smaller branches than the astrocyte. So it's sort of a similar shape, but just the branches get smaller and smaller, and it's a smaller cell overall. But like the astrocyte, it's attached to the neurons. Their function is to be a part of the immune system in the brain. So stay tuned for another video that I'm going to include in the module about these cells and their role in chronic diseases and even in COVID-19. So it's very timely and relevant. Panel C on the left, we see ependymal cells. These line the brain and spinal cord tissue, sort of like a skin, mostly in the brain's ventricles. And what they do there, especially in the ventricles, is secrete cerebrospinal fluid. This is the fluid that circulates all throughout your brain and around your spinal cord to insulate and protect those structures. It keeps your brain from scraping up against your skull and such, and your spinal cord from getting injured. It's a little bit of insulation and kind of shock absorption. Then on the right, in panel D, we see an oligodendrocyte, which we also saw in the electron microscope, but it looks really different on this slide, and that's partially because what we were looking at before was a cross section, where if we looked at one cut in half and looked at that sliced part, here we're looking at it more whole from the outside. What they do is they have a nucleus, and then their cell body makes a very thin layer. Instead of just being kind of a circular cell with some branches like most of these are, it takes its cell body and grows this really long appendage that wraps itself around like a sheet, and it makes this like fatty layer of a sheath around part of an axon, a nerve fiber, or part of a neuron that we can call a nerve fiber or an axon. It's this long electrical cord-like portion of the neuron that sends messages. We'll talk more about that later, but this insulates that electrical cord, basically, is what the oligodendrocyte does. And the Schwann cell in panel E at the bottom actually does the same thing, but it does so in the peripheral nervous system. These cells are just called something different when they're in a different part of the nervous system, and they're shaped a little bit differently, but their function is the same. We also have radial glia, not pictured here, but they secrete chemicals that help guide neurons when they're developing. Remember I told you that that's part of a neuron's life cycle. They have to migrate soon after they proliferate and start differentiating into a neuron. They follow the chemicals sent by radial glia, and when that process is disrupted, you can have some disorders, like schizophrenia seems to be linked to genetic conditions that cause differences in this migration process and in these radial glia. So we talked about these supporting cells, but what about the neurons they actually support? They can have a lot of different shapes based on where they are in the nervous system and their exact function, but we're going to look at a typical template for them here. On the left, we have this blobby cell body with a nucleus in the middle, kind of like other body cells that we have. It looks sort of like the astrocytes we've been looking at, with the starfishy arms coming off of it in most directions. And these branchy extensions off to the left are called dendrites. They receive messages from other cells. They receive chemicals actually from other cells, and then they start conducting an electrical message that they send to the soma or cell body. And the soma processes this message some more, and then it sends that electricity down the long snaky arm of the axon that we see moving off to the right of this image. The axon ends in really small protrusions, little kind of fingertips, that can be called axon terminals, terminal buttons. Your textbook says bouton, French word for button, which I think just sounds kind of silly and pretentious, or a variety of different names. This is the sending end of the neuron. The cell body, after processing its messages, transmits it down the axon all the way to the axon terminals, and then the axon terminals or buttons or whatever send a chemical that will be released that then communicates to a neighboring neuron. It will be received by another neuron's dendrites, and the process continues. The axons of many neurons are insulated with a sheath made from those oligodendrocytes that we just looked at, and Schwann cells in the peripheral nervous system. Each of these cells, oligodendrocytes or Schwann cells, each has their own cell nucleus, and then they make this kind of sheet of fatty material that stretches around the cell body, and they stretch their cell bodies around the axon in this kind of spiral pattern. So you have multiple layers of insulation. And that's actually what it is. It helps the axon conduct its electrical signals without kind of leaking some of that energy outward. It's sort of like insulating a wire with rubber, so it becomes a cord or a cable, and you're not going to lose any of that electricity to any materials to the outside air, or to touching it and getting shocked, for example, or kind of like wearing a jacket to keep your body heat inside and near your body. And several oligodendrocytes or Schwann cells in the peripheral nervous system will live on a single axon, and there's tiny gaps between them. These gaps are called nodes of Ranvier. It's a French name. It looks like Ranvier, but it's Ranvier. The electrical signal kind of jumps from node to node, and we're going to talk more about this process later. These neurons are constantly prepared to send a signal, even when they're not currently receiving or sending any messages. They're ready to go. They're just pent up with this potential, kind of like a rubber band being stretched back and ready to be let go. So on the next slide, we're going to start looking through exactly how that potential energy is stored, and then also later how it's released. So when we talk about how it's stored, this is called resting potential.